Okay, hello everybody. My name is Miss Pearson, and today I'm going to go over DNA structure with you. Before we start the slides, please take some time to read this article before we begin the notes. Just copy that link into a new tab, hit pause on the slideshow, read that article, and then when you're done, come back to the PowerPoint, hit play, and we'll go through the DNA structure notes. All right, so DNA, what is it? It is deoxyribose nucleic acid. So here we've got our D in DNA, deoxyribose. N is for nucleic, and A is for acid, DNA. So there's 6 feet in every cell. It's composed of nucleotides, and it has 2 primary functions. So function 1 is to control protein production, and number 2 is to duplicate itself for new cells that are created. That way all of your cells have DNA in them. Your DNA will also undergo changes, and these are called mutations. So some vocab for you. Chromosomes. These are tightly compacted DNA. So in this picture, boom, there's our chromosome. So they're only in X or Y form during cell replication. In humans, we've got 46 chromosomes. And interesting fact, chickens, they've got 78 of these things. And strawberries, they have 14. Another vocab word, chromatin, which is unwound DNA. So here we've got our chromatin. It's unwound DNA, right? And then if you look at the rest of this picture, here's the double helix that we might be familiar with, that structure there. Some more vocab. Genes, they're a segment of DNA. They control the protein making in a cell, and they are made up of nucleotides. So if we were, for example, to look at this segment of DNA, and that was our gene that coded for, let's say, blue eyes, then yeah, that would be that segment made up of nucleotides. And so here's our nucleotide, and then our genes are on a chromosome, which again is this. Okay, so where is DNA found? Well, if you recall from our cell unit, right, it's in the nucleus of the cell. And every human body cell, except for the egg or the sperm cells, they have 46 chromosomes. So you get 23 from your mom, and you get 23 chromosomes from your dad. And together, to make you, get 46. And then nucleotides, these, if you recall from our macromolecule unit, then these are the deoxyribose sugar, so this guy here, our phosphate group, and our nitrogenous base. And we've got 4 nitrogenous bases, which we'll dive into in this next slide. So again, our nucleotide is composed of 3 things: a sugar, a phosphate, and a nitrogenous base. And those 4 bases are adenine, thymine, cytosine, and guanine. So we've got 2 types. We've got purine, which is a double ring. So our adenine and our guanine are purines. They've got 2 rings. And then our second type is the pyrimidine. It's a single ring, and those are thymine and cytosine. So we've got one ring each for those. And there's this thing called Chargaff's rule. So your amount of guanine will be equal to your amount of cytosine. And then your amount of adenine will equal your amount of thymine. So here we've got, here we've got C's equal G's and A's equal T's. So in this little database picture, Homo sapiens, us, got about 31% A's and roughly 31% T's, about 19% G's and almost 19% C's. So it's not perfectly equal, but just about. So rule of thumb, you can just know that G's and C's are equal and A's and T's are equal. So who discovered the structure of DNA? Well, through X-ray diffraction, Maurice Wilkins and Rosalind Franklin discovered its structure. So in this photo down here, they had this X-ray diffraction of DNA and they saw that it was a double helix. Also, this is Rosalind Franklin. Okay, so with that information, James Watson and Francis Crick, knowing that DNA is a double helix, which is kind of like a ladder, but it's just twisted. So here's our ladder version, and there's our, left and right. And there is our twisted version. So they found that a sugar and a phosphate form the backbone. So here we've got our sugar, and then this circle is our little phosphate. So that is what makes up the backbone. And in between our backbones, we have the bases here. So there are bases, which are the nucleotides, A, T, G, and C. And those are bound together with hydrogen. So if we jump over to the twisted version of our picture, our A's and T's have 2 hydrogen bonds, and our G's and C's have 3 hydrogen bonds. So here's a picture of Watson and Crick with their DNA structure that they put together. So we've got our sugar phosphate backbone here, and then in the middle, all those nucleotides. Look at him admiring his beautiful work. Okay, so one of the characteristics of DNA is that it's complementary. So complementary in this scenario means that bases on one strand will match the bases on the other strand. So A's and T's bond together and G's and C's bond together, as we saw in this picture. We've got our A's and T's and our G's and C's there. So if you have a strand that's A, T, G, 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 C, and C, T, A, your second strand will be T, A, C, 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 G, G, A, T. So if we were to write our own strand, we had, let's say, T, A, G, C, A, T, excuse me, knowing that A's and T's go together, what's going to go with this T? Be an A, right? And what's going to go with this A? A T. And knowing that G's and C's go together, we can know that this will be a C. And so same thing with this one here. We've got a G, a T, and an A. So these are our complementary strands. And DNA also has a very specific orientation. So nucleotides will form this chain here, and the phosphate end, here's our phosphate, is 5', and then the opposite end is 3'. So if DNA is complementary, what end is going to match the 5' end? So here's our 5', and if we go across, it matches the 3' end. And then if we go back to this first strand, the bottom by our sugar is the 3' end bound to the 5' end with our phosphate there. So strand 1, strand 2, you've got your 5' end with your 3' end, and on strand 1 your 3' end, then to strand 2 will be your 5' end. It's where those line up. So if we compare different DNAs, we've got prokaryotic DNA and eukaryotic DNA. So in prokaryotes, our DNA is double-stranded. It's also circular. And there's one chromosome, and that DNA is in the cytoplasm, just loosely floating in there. And then supercoiled DNA is called a nucleoid. And the prokaryotic DNA does not have histones. But in eukaryotic DNA, it's also double-stranded, but it's linear, right? So it's got a beginning and an end, and it's usually at least one or more chromosomes. Again, humans have 46 chromosomes, and our DNA is found in the nucleus. And then chromatin is DNA that's wrapped around the histones, which are a protein. So again, prokaryotic DNA does not have histones, and we do have histones, so our DNA is wrapped around that. And that is what makes up the chromatin. Okay, so back to the prokaryotic DNA. Prokaryotes have plasmids, which are small circular double-stranded DNA molecules, and they're different from the chromosomal DNA. They will provide the bacteria with genetic advantages, for example, antibiotic resistance. So each daughter cell, let's see here, will receive a copy of a plasmid. So here's a plasmid that could go to a daughter cell, this could go to a daughter cell. And bacteria can also transfer the plasmids to one another through conjugation. Okay, so that was the bulk of DNA structure. So let's go back to that National Geographic article. So DNA from Stone Age chewing gum. Where did that DNA come from? If you read the article, you might be able to remember where it came from, and if you read it, you should be able to answer most of these questions and come up with some ideas. So is that DNA the same as our DNA? What could be different about her than people today? What causes those differences? What did they learn from her DNA? How do you think they extract DNA? And did they find DNA of other things from that chewing gum? So based off of what we just went through on these slides, what information we just learned, and based off of that reading, what kind of connections can you make to answer these questions? So what I'd like you to do is actually put together a Flipgrid. Get familiar with Flipgrid and spend, like 30 seconds or so answering some of these questions in Flipgrid, and just take a look at that and what your peers are saying, things like that. But otherwise, for Wednesday, if you have questions about anything, about the material, the article, or how things work with online learning, like how is Google Classroom running, how do you use Flipgrid, stuff like that, Let your teachers know, send us an email or get in touch with us through Google Hangout. So we'll also be sending out a worksheet, so you have access to that on Google Classroom, and it'll be due Friday. And a reminder that office hours are from 9 to 11 and 1 to 3. So if you need something, please get a hold of your teacher during those times. Your teacher might have specified that they only have biology office hours at a certain time, maybe from 9 to 11, or maybe they have bio from 1 to 3. So just be aware of that. Make sure you know when your teacher has office hours. But again, if you have any questions, please reach out to your teacher. If you need help with stuff, if you need some support with things, we're happy to help, and we're here to support you. And during this time of not really knowing exactly how distance learning is going to go, we're all in this same boat, right? So we're here to support each other and make sure we all kind of have our ducks in a row and we're helping one another. So yeah, if you have any questions, again, please reach out. Otherwise, thanks for going through these slides. All right. Bye.